Okay, so cannibalism, mad cows, and yeast. Hopefully I'll provide a link with that, and that link will be prions. The Australian government in the 1950s carried out a census that encompassed the population of Papua New Guinea. And within that census, they realised that there was a neurodegenerative disease called kuru that was endemic in population of people who live in the central part of Papua New Guinea called the foreign people. Ritual cannibalism, the rules, the men get the juicy bits, the women and children get the not so juicy bits. And one of the problems here is that the not so juicy bits contain the infectious particles. These are some women and children with kuru. This is a sheep with scrapie. People realised that scrapie is a disease of sheep that's been around since the 1850s. These collectively have similar symptoms and pathology. They collectively are called the transmittal spongiform encephalopathies, TSCs. Note in humans, Creutzfeldt-Jakob disease and variant Creutzfeldt-Jakob disease. Variant Creutzfeldt-Jakob disease has been acquired when someone has eaten infected beef. And in cattle had broken the species barrier with regards to this cross-contamination of this BSE disease. Okay, this guy, Stanley Pussner, won the Nobel Prize in 1997 for coming up with his discovery of prions. Prion stands for proteinaceous infectious particle, and he realized this was the infectious agent in these diseases. What is a prion? A prion, like I said, infectious protein, doesn't have any nucleic acids, DNA associated with it. This is a heretical idea in biology because the flow of genetic information goes from DNA to protein, transcription, translation. Pussner suggested that proteins could contain genetic information. What happens is a protein called PRPC, this is the prion protein. This is how it folds. It changes shape. When it changes shape, it becomes infectious. It forms fibers. These fibers deposit in the brain. You have neuronal cell death. It's not replaced. You have neurodegeneration. That's all I'm going to say about mammalian prions for now. You can read further. There's a recommended book to you called Deadly Feast by Richard Rhodes. It's a good read. It can be slightly soap opera-ish at parts, but that's I enjoyed it. I'm not sure what that says about me. Yeast has prions, so I work with yeast, Saccharomyces cerevisiae. A guy called Brian Cox is a yeast geneticist, identified a strange genetic element, which he called psi. 30 years later, a guy called Reed Rickner realized that this psi factor was a prion form or a protein called sub-35. Don't get confused here. I'm not saying that there's a mad yeast disease that exists or that you can catch prion diseases from eating bread or drinking beer. What I'm saying, there's similar mechanisms in proteins in yeast that can fold and become infectious. This means that we can use yeast as a model organism for studying human disease. Why is this possible? Because many genes in yeast are similar to genes that we have and produce similar proteins. Don't take my word for that. You can take the word of the Nobel Prize Committee, who gave the Nobel Prize to these two guys, Lee Hartwell and Paul Nurse, in 2001. Both of these guys work on yeast systems, and they're fundamental discoveries of the cell cycle that are related to cancer. Okay, so they work on yeast, but they got the Nobel Prize for medicine. So the relationship is there. How do we monitor yeast with prions cells? These white stroke yellow looking colonies that we get, these contain prions. The red cells down here, these do not contain prions. This means that we can carry out quite simple genetic screens to look for cells that we can make turn from white to red. Okay, we use a combination of genetics, computational biology and biochemistry to work out the functions of these proteins that are involved in prepropagation. Yeast can also be used to screen for drugs that cure prions. This is an agar plate that contains millions and millions of yeast cells. These discs contain drugs. Where they're red, the prion is being cured by these drugs. Drugs that cure yeast prions can also cure mammalian prions. Yeast prions can be beneficial as well, so yeast cells that contain prions are more resistant to stresses than yeast cells that do not contain prions. So stresses such as heat shock or DNA damage. This has massive implications from an evolutionary perspective. Can you get beneficial prions in humans? It appears you can. There's a protein called MAZ, which is involved in viral signaling in resistance to viral infection, and also a protein called CPEB, which is involved in memory. They also appear to be prions. Two things to finish up on. First of all, prion diseases. If Santa acquired a prion disease, the fact is he would die, even if it came through consuming infected beef, such as Rudolph, or in terms of spontaneous infection. Frankly, he would die. But the point I want to make to you is that if Santa looked through the scientific evidence, what he would say to you is that, yes, prions exist, they cause disease, but perhaps not all prions are bad, and they may be even beneficial to some cells. don't associate the term prion with just disease.